All right, so let's continue on to protein creation, starting with transcription. So the process of transcription occurs within the nucleus of a cell. It's broken down into three stages: initiation, elongation, and termination. So at the first stage of transcription, which is initiation, an enzyme known as RNA polymerase is binding to the promoter region of the DNA. So as we can see here, this is the original DNA. So clearly, RNA polymerase, which is this Y-looking thing, has binded to it, and they start unzipping it. So once the RNA polymerase unzip the DNA, it makes the template strand of the DNA become exposed, which makes the RNA polymerase able to add complementary RNA nucleotide to the template strand of DNA. Please remember the nucleic acid RNA has different nitrogenous bases to DNA. So U has replaced T in DNA nucleotide. That process continues. So this newly formed strand of mRNA elongated, and this is when pre-mRNA is created. So once the full area of the gene is transcribed, which is when there is a stop codon and that codon has been reached, transcription will stop, which is termination. So during termination stage, RNA polymerase will detach from the DNA strand. But this process doesn't just stop there, because there is still a post-transcription modification that needs to take place. So this process is also known as RNA processing. Three very important things happen during this process. One is introns are removed. So if you can recall, introns are the non-coding region. And exons, the coding regions of DNA, are stuck together, which is also referred to as spliced. So they splice together to create mRNA. The second thing that happens during post-transcription modification is the attachment of poly A tail. So this is attached. We're not going to go into too much detail about what is really poly A tail. You just need to know it's a little molecule that's attached to the 3' end of the mRNA. On the other side of the mRNA, which is the 5', a methyl cap is also added to make the mRNA more stable. So we often refer to these as a methyl cap, but just for your information, this is a very special guanine that has been methylated. Okay. So both these two structures, the poly A tail on the 3' and the methyl cap 5', are added to prevent damage and degradation of the newly made mRNA. So prevent damage and make the whole structure more stable. Okay. The other thing that is not mentioned on this slide is alternative splicing. So this is a special process that occurs in both prokaryotic and eukaryotic cell. So by selecting or selectively including or excluding certain exons from pre-mRNA, this mechanism, so it's referring to alternative splicing, expands protein diversity and number of proteins that can be encoded by genome. So by doing these, we can see that proteome is often or always greater than genome. Okay, now let's move on to translation. So different to transcription, translation occurs in the cytoplasm. So at this stage, mature mRNA has moved out of the nucleus and into the cytoplasm. So once it's in the cytoplasm, it will interact with ribosome, which is actually made of another type of RNA. So rRNA is the main component of ribosome. rRNA interacts with mRNA and help mRNA to start constructing a polypeptide chain. So if you can recall, polypeptide chain is when multiple amino acid joining together using peptide link. So don't worry too much about the subunits of polypeptide chain, but this is important. So every time the mRNA bases are read by 3, so 3 mRNA bases is translated into 1 amino acid. So what happens is tRNA, which is short for transfer RNA, it's another type of RNA. This special type of RNA has amino acid attached to one end of the RNA, and the other end, it's got a region that has anticodon, which means it's got 3 bases that's complementary to the code on mRNA. So every 3 bases on mRNA is called codon. So tRNA will bring appropriate amino acid that correspond to the mRNA triplet by a codon-anticodon relationship. Just for information, again, this is beyond our curriculum. Usually after proteins are first made, there's post-translational modification as well. So similar to what happened before, this modification is to increase proteome diversity. So depends on how they are later processed and folded, proteins can increase their diversity as well. Okay. Now, please make sure you know how to read and use the amino acid table. Special thing about DNA code is that they are considered degenerative or redundant. So these 2 words are the same, so they're equivalent, and they can be used interchangeably. So what these 2 words both mean is that there's more than one DNA triplet / RNA codon that code for a specific amino acid. So let's take example of serine. So serine, these type of amino acid can be coded by 4 different types of code. Okay, so these 4 codes will all produce serine as a result. So the benefit of these, the redundancy and degenerativeness of coding, provides a level of safety so that not all mutations result in abnormal proteins being created, because that can be a big trouble for the organism. All right, to sum it up, make sure you can compare and contrast between DNA and RNA. You should also be able to distinguish different types of RNA. Type of RNA you need to really focus on is mRNA, and also the difference between pre-mRNA and post or mature mRNA. All right, so that's all the content covered. Let's now have a look at a few questions. Question 7. The following diagram outlines the process that occurs in living cells. So without looking at the questions below, I'm just going to have a look at the diagram first and then sort of try to interpret the diagram. So what I'm seeing here, based on the labeling, so this dotted line looks like a partial nuclear membrane, which indicates that this must be within the nucleus. All right. So process X depicts that a double-stranded molecule turning into a single-stranded molecule. And that should ring a bell because we know double-stranded molecule, and based on the shape of it, this must be a DNA molecule, and it turns into a single-stranded molecule, which must be RNA. Okay, and it further confirms our prediction because this entire process happens in the nucleus. So process X must be transcription. Okay, now mRNA has been made and it travels outside of the nucleus into the cytosol. And then we can see it's attached to a circular thingy, which also has got two parts. So this is a typical drawing of a ribosome. Okay, so ribosome consists of two subunits. Don't worry too much about the details of that, but it just helps us to recognize these two things that make up, the structure that made up of two things are ribosome. So mRNA is attached to ribosome, and ribosomes start helping the translation process. Okay, so process Y looks like translation. So this structure here is a typical drawing of tRNA because we know tRNA has got one side that's got an amino acid attached to it. And on the other side, it's got a 3-base anticodon that should be able to match a codon that's on mRNA. So once anticodon and codon forms a pair, tRNA will help dock the amino acid at that certain location on the mRNA. Okay, now let's have a look at the question. Question A1, name the process represented by X. So based on our interpretation of the diagram, X is transcription. Okay. Question 2, describe the sequence of event that occurs during the process at X. So given that X is transcription, there are 3 steps involved in transcription, which are initiation, elongation, and termination. Okay, so I'm not going to give you the answer straight away, but instead I'll put in a few keywords. So please give this question a try and include the keywords I've listed here. All right, so moving on to question 8. The following diagram shows some of the steps in the production of a protein within a cell. So given that this is protein production, we are probably looking at transcription and translation. Okay, so let's have a look at the diagram first. Now the first part of the diagram shows us a DNA segment, which is a gene. So this particular part of the DNA must be containing the code for the protein that we're trying to make. So step 1 looks like DNA, the double-stranded molecule, has turned into a single-stranded molecule. So once again, this process is transcription. And then looks like there's not much change between molecule E and F. So let's have a look at what are the subtle differences. So the coding looks like it's the same, but what's different is we have a small circle attached to the 5' end of the molecule and a chain of A's attached to the 3' end of the molecule. Okay, now you probably realize this is post-transcriptional modification. Okay, if you don't want to remember the really long name, it's also referred to as mRNA processing. And remember, there's 3 things involved in the process. So first is the addition of a 5' methyl cap. Second thing is the addition of 3' poly A tail. And then last but not least, which is step 3, is splicing. So as you can see here, it looks like all the white regions have been removed and only the black regions remained. So this is a clue for us that white region must be the non-coding region because they have been removed. And the name for non-coding region is intron. So the black region must be the coding region. They have been spliced together. And the name for coding regions are exons. Okay, now let's have a look at the options below. Which one of the following is a correct statement? So A says step 1 represent translation, which is incorrect because it should be transcription. Question B, there are 6 introns in the gene. So introns is, as we figured out before, the white region. So let's have a look at the molecule E and then count how many white regions are there. So we got 1, 2, 3, 4, 5 different white regions. But the option suggests that there are 6 introns, which is incorrect because we have just counted and there's only 5 introns. What about C? RNA polymerase is required for step 3, which is not true because step 3 is splicing. Splicing requires the help of spliceosome. And RNA polymerase mainly facilitate the process of transcription. So it's helping to binding to promoter regions, bring complementary nucleotides, yada yada. Okay, so our final hope is D. The circle on molecule F represents a modified guanine molecule, which is true. Okay, so the modified guanine molecule is the 5-methyl cap, and we have formed that methyl cap by modifying a guanine or methylating a guanine. Okay, so option D is indeed correct. All right, moving on to question 9. So question 9 says different cells within an organism have different proteins. In some cases, different proteins can be coded by the same gene. Okay, so it's saying the same region of DNA, which has the same code, but all the code, when they're transcribed and translated, can produce different types of proteins. So the question asks us, explain how expression of a single gene can lead to the production of different proteins. Okay, and this is a 3 marks question. So let's plan our responses first before we actually start answering and put them into writing. Okay, so this question is asking how do we have limited genome but then also have the ability to produce a more diverse proteome? Okay. The key is alternative splicing. Okay. So already I've assigned a few marks to the points I want to include in my answer. So number one, so the first mark goes to limited genome produce a more diverse proteome. Number two is the definition or description of the process of alternative splicing. And the third mark will go to the result or the product of alternative splicing. Okay. So once again, I'm not going to give you the answers. Please give this a try based on the mark breakdown and allocation I have suggested here. And yeah, if you got any questions, please let me know.